Good evening, everybody. How's everyone doing this fine Thursday evening? It's nice, Friday Junior. Woo woo. All right. Welcome to the 2024 celebration of undergraduate student research. Before we begin, I'd just like to have everybody give everybody a round of applause for all of our hardworking student researchers, please. Thank you, thank you. Let's get into it. Here's our agenda. We have remarks from Dean Hazel Siv and Dean Rochelle Risberg. And we have our presentations, and then we have a little dessert and networking time at the end. Now let's get into it. My name is Dante Lewis. I am a third year cell molecular biology major. Currently, I'm on co-op, which is very fun. I work at Alnylam doing quality control. If you asked me what that was when I got hired, I wouldn't tell you, but now I can tell you all about it. I am a COS student. I'm the vice president for Student Leadership Council for the College of Science. I am the president of the biology club, and I am a community service office trip leader and volunteer. As far as my research background, I have won 2 Peak Awards for addiction and all facets of addiction. And just a little bit about the future, continuing research in the Civ Lab, and hopefully a biopharma regulation and policy co-op as for my next co-op. My research award is from the Dean's Fund, the Dean's Undergraduate Research Scholar, and my PI was Brandon. All right, and we are ready to get into these presentations. So we have our remarks from our Dean, Hazel Siv, who is the Dean of the College of Science and a professor of biology.
Lovely. Let me just get organized here. Oh, that is a really. I know I'm little. We need to grow. Okay. You have a little Dean. It's lucky I have a loud voice. Make up for that. Well, this is lovely. This is absolutely, genuinely a wonderful event. I am absolutely thrilled to see everyone here. Maybe you can raise your hand when I say your category so we can kind of know who everyone is. So I'll start with alumni. Wonderful. Families. Wonderful. Friends. We love friends. Terrific. Our supporters. Thank you. So good to see you. Some of our very eminent supporters. We'll point out in a moment here. Faculty. Yay. Thank you so much for coming. It's just fantastic to see you all. Postdocs. Thank you. And of course, students. Yay. A round of applause for everyone. Thank you so much for coming to this wonderful celebration of undergraduate research. I'm a scientist, and I began my own research career as a first-year zoology and chemistry undergraduate major. I had been working since I was 14, and it was, wherever I could find a job, it was very often in clothing stores. And I'm really good at folding sweaters. I'm just, like this. I'm really good at it. But it was totally not so interesting. And so when I was a first year undergraduate, I decided to see if there was a more interesting job on campus. And this was way across the ocean in South Africa, where I'm from and where I got my undergraduate degree. There was no undergraduate research program. But I thought, maybe. And so with my heart knocking, this is for undergraduates who think, that no one else is scared except you. My heart was beating, and I went to the chemistry department, walked down the corridor where the doors were all black painted and they had frosted glass windows. So you had no idea if there was someone in the office or, whatever, what even went on behind the doors. It was kind of scary and magical at the same time. And I went to Professor Perol's office and I knocked at the door and he was there. And I went in and he said, "How can I help you?" And with my heart, beating away, I said, "I'm a chemistry student and I was wondering if you have any work for me." And he said, "Sure." And I thought, whoa, amazing. So I went into his lab that summer and I did thin layer chromatography. And that kind of just set me on my way. And even though there wasn't an undergraduate research program, now I felt empowered. And I just went on every vacation or every summer and found another professor to work with in the zoology department, etc. That's how I started. And I thought research was really interesting. I think science is really interesting. I absolutely love science. Science research is fantastic, and we'll talk more about that in a moment. But I want to tell you that as a research leader of my own group, I've had hundreds of undergraduates do research in my own lab. Some have been there for just a short time. Some have joined right when they came to the university and have stayed throughout their career. It's a mix. Here at Northeastern, where I moved from MIT 4 years ago, I set up a small research lab in the basement of Migue. We have frogs and we have fish, and we do really fascinating research that looks at development of the face and the brain. We're really interested in trying to model neurodevelopmental disorders using our zebrafish and move towards the beginnings of novel therapies. It's really important, fascinating research. I'm very proud of it, and I'm thrilled to continue it here at Northeastern with undergraduates in our lab since I've been here. And Dr. Brandon Chu over there is our postdoc who really is doing a wonderful job supervising a number of students. We've had Dante, we've had El Kim Levesque. I'm not sure if there you are. So good to see you. And we've had a number of others. So it continues. For me, the commitment for undergraduate research is personally very strong. From the platform of me as an undergrad, me as a lab head, and as an innovator in higher education, I've built a very large higher education initiative. I've taken groups across the multiple continents talking about problem-solving approaches to education, teaching curriculum design workshops. I've really been in the higher education space for a long time, and I can tell you that I'm very confident in saying that undergraduate research is extremely important. It's a really important part of any student's trajectory, no matter what your future path. You don't need to do undergraduate research because you want a career in research, although we'll discuss if you do, that's great. But for anyone, undergrad research is fantastic. And I'm a huge fan, as you can tell. In that view, promoting undergraduate research is a goal that I set for the college when I joined as dean. It's a key goal. One has many moving parts, but supporting the students through funding is absolutely crucial. And I'm thrilled, thanks to you, that we have made excellent progress in this goal. Much, as I say, of the progress is thanks to our supporters, you, alumni, families, friends, trustees emeritus, for the investment and philanthropy that enables exceptional research experiences for undergraduate students. And you may know or not that we are celebrating 115 years of co-op at Northeastern. This university started on co-op, on work to get through university. It's continued the wonderful tradition, the so important tradition of co-op throughout these more than 100 years. And this celebration, we can kind of interdigitate with that wonderful landscape. So I want to talk a little bit more about some aspects of the importance of undergraduate research, both for the undergraduates, but for everyone else. But I want to also take a moment to extend enormous appreciation to everyone who has allowed us to create the New Dean's Undergraduate Research Award. Alumni, families, friends, the contributions are fantastic and they are so important. And many of you who are students have benefited from these terrific gifts that have enabled your summer programs, your summer research, or research at other times. I'd like to give a special thank you to Carol Shapazian. It's so good to have you here, Carol, for her important support of undergraduate research experiences through the Carol Shapazian Research Co-op. And I know you are here, Bita. That's very good to see you. And you are here. It's your
I'm the PI.
You're the PI that is
Yes.
supervising. It's a pleasure to see you. We will meet properly in a little bit, but it's so good to have you here. So thank you so much, Carol, for your ongoing support. It's an honor. A heartfelt thank you to the Halverstadt family who are so good to see you. Thank you so much for being here. Um, the Halverstadt family has created the Family Summer Internship Program that has given absolutely wonderful opportunities to multiple students. And we're so grateful for your ongoing support and engagement. It's a pleasure to see you. So it's a pleasure to see Anne when she joins us. And we're so proud of her as one of our alumni. So thank you so much. Thank you to all the faculty, past and present, and I want to highlight Professor Paul Vouros for your contributions and for a naming of a co-op fellowship in your honor. It's such a pleasure to have you here, and Irene, and we are delighted to have you join us this evening. Thank you so much. Thank you all. Thank you to people who are not here, who are online. Are we online? That was so seamless. That's very impressive. If you are here online, such a pleasure, and thank you so much. Let's give a round of applause, please, to our supporters for these. You know, research is the closest thing to magic. It's the means to discover what has never been known before, what has never been uncovered in the universe before. Wow. Research in science is particularly magical because all science research starts with discovery. In the College of Science, we make discoveries from the tiniest to the largest, from the great language of the universe, mathematics, to the tiny fundamental particles described by physics, to the molecules of chemistry. If you want, you can sort of stand up and wave for the discipline when I call you. You know, I didn't tell you that, but you are, I know, cheerleaders for your majors and wonderful. They're all fabulous. Shall I start again? And then the undergraduates. Okay, I'll start again. Okay. From the great language of the universe, mathematics. See, yay. To the tiny fundamental particles described by physics. Fantastic. To the molecules of chemistry. Cells and organs of biology. I bet that's a bunch of you. Yes, you're a little shy there. The mind of psychology.
Yeah.
Yay, all those neuroscience students, I know. The great ecosystems of marine and environmental sciences. And the cosmos, again, in physics, we go from tiny to the largest. Discoveries in science are often made by bringing two fields together, and the applications of them are generally collaborative and cross-disciplinary. Fundamental discoveries of how things work are always so interesting. I'm looking at the faculty around. Dr. Monahan, chair of biology, is just right in my line of sight. So good to see you. So I look at you, Dr. Apfeld in biology, and then, you know, there are others sort of there. Thank you so much. It's wonderful to see you all. A lot of marine environmental science up in that corner. Yeah, okay. It's Dr. Garcia, Dr. Lara, yes, Dr. Coley, who did I miss?
Dr. Gates.
So good to see you. Remy, I don't know that we've actually ever met. You know, it's so good to see you. Yes, it's great. So Dr. Gayton is one of our newer faculty members, also from Marine Environmental Science. What a pleasure. Discovery is what we do in science. It's fascinating. It's what you as undergraduates should be doing. But discovery always, and I really mean always, turns into application. It may take decades, it may take hundreds of years, but every medicine, every new therapy, every understanding of new energy sources, every understanding of how climate change works and strategies to preserve our planet, every understanding of the universe, all of these things come from science research, and all of them turn into discoveries that help people, that help the planet. It is an extraordinary mission that we have here in the College of Science, entrusted with knowledge, entrusted with using knowledge, many times way in the future, but always. Undergraduate research is hugely important in our mission. My long-term goal as dean is to enable every College of Science student who is interested to try some research. And I want to explore just for a moment why undergraduate research is so important. This is for the undergraduates as well. You might be having fun, but actually there's a whole lot more that I'll just, tell you, and you can do what you want with it. For some of you, this will be a future career. Research will be so interesting that you'll decide to put your time and your talent into research, and that's wonderful. To add to the research considerations of the United States and the world, the mission, hugely important, whether you are in industry or academia. But every student, regardless of your future career, whether you go into medicine and you never do any more research, whether you go into finance, into communications, into education, into management, into policy, whatever you do, regardless, your careers will benefit from immersion in the research experience. Students understand, you understand the relevance of concepts, hands in, what you learn in the classroom, now you're applying in the research. You're exposed to real life problems, and they're often across disciplines. They're not in one small track, they're cross-disciplinary. You may understand something about innovation and entrepreneurship, what you can do with the research. Some of you may be doing actually applied research, and some of you may think, what could I do with this research? And that's really important. And the huge point that every student, all of you, will gain skills and confidence in solving problems. The problem-solving acumen that you will get doing research in science is unparalleled. You will put it in your pocket, you will put it on your CVs, and it will be with you always. It will let you understand what formulating hypotheses are. Those of you pre-med, who's pre-med? Maybe. You don't have to know, it's fine. Those of you who are going into medicine, you will know how to figure out what's wrong with someone by formulating hypotheses and figuring it out. You'll be able to construct answerable questions. You'll learn about how techniques can help you answer questions. You'll develop skills in data analysis. You'll learn not to trust data. You'll learn to listen hard to your data. I call it let your data whisper to you. You'll learn to understand what you can believe, what you can't believe, and how to actually do reliable, valuable research. You'll learn about failure. The failure doing research is probably perhaps the most important lesson. Nothing ever works, really. And then you do it again, and your experiments work, and from when the techniques work, you learn something. And it's a valuable lesson that you keep going and you keep going and you keep going, and it's okay. And learning to communicate. What are you learning? Why? What if you think about what you've learned, how to communicate? All of these skills are so important. Every single one of them will help you now and in your next career steps. And then finally, our College of Science gives undergraduate researchers even more. We have in the college defined a unique, important landscape that we term the good power of science. The good power of science means that our students, the next generation of science professionals, no matter what you do, are trained to act with a responsible, ethical mindset. It means our research in the college addresses crucial societal challenges and is framed with an ethical, responsible commitment. Everyone, regardless of their position in the College of Science, is charged to act with ethical, responsible, respectful principles. That is the good power of science. You're part of it. It's a wonderful mission. It's a wonderful way to engage with the landscape of science and with the research that you're doing. I am very proud of this landscape and very pleased to have every student part of building the good power of science. I want to welcome everyone to this great celebration. Thank you for being here. Congratulations to everyone involved. Now I'm delighted to introduce Assistant Dean Rochelle Reisberg, who leads us in many undergraduate program aspects and who has been an absolute pioneer and champion for undergraduate research in the college. Rochelle, thank you so much.
Okay, thank you. Well, I am just so passionate about the research mission, the ability to impact young people and see their trajectory is just so close to my heart. I've been at Northeastern for about 20 years, and when you have a student come back after they've had these opportunities, they finished their PhD at Stanford, they finished their PhD at Johns Hopkins, they finished their PhD at MIT, they finished med school, they finished their PhD at Oxford, and they come back and tell you their first year, their second year here as an undergraduate, the ability to be in a research lab and be entrusted and mentored made all the difference to them. And that is what we're all about. We want to bring these opportunities to our students as early as possible and provide as many of them as possible so that our students can flourish. We had a freshman research meeting today and actually last week, and a number of those freshmen are in this room. They're starting their trajectory in research. We invited some of the sophomores in, and so I see some of those sophomores are actually here. When they stood up and explained what they had done in 1 year, the freshmen were just in awe. And all I could say was, yes, that will be you in 1 year. It really will. So to the students here, congratulations. You inspire all of us. You inspire me. To the faculty, thank you for the opportunities you provide. Thank you for being patient when we knock on your door and ask for one more spot. Can't we fit one more student in? And to the folks who've made this financially possible, that makes all the difference because many of the students really cannot do these types of, cannot take advantage of these types of opportunities if they didn't have the financial support to make that happen. So I'm going to turn this over now to our MC, but I just want to say something about him. So you can't hide back there, Dante. Come on, come on over here. So I met Dante 1.5 years ago. Yep. Coming out of the a program and a Torch Scholar, which, if you know anything about that, it's a remarkable program. is a remarkable young man. And he is, in some ways, the model of what we can offer to our students. And he's the type of student that you ask him, would you like to come to this meeting? He says, yes. Would you be willing to lead this? Yes. Could you be the MC for this event? Yes. So to the students here, say yes to these possibilities, because that's how you're going to grow. That's how you're going to take it to the next level. So I turn it over to you, Dante. You're going to lead us through what is almost like a piano recital for us. Thank you.
Oh. Bring this up just a little bit. I'm sorry. Yeah. Thank you again for the remarks given by Dean Reisberg and Dean Siv. Wonderful, wonderful words. I'm inspired already, and I still do research. So now we're going to pivot and start focusing on our wonderful students that have volunteered, I think, to give presentations today. So we're going to start off. Our first one is Tochi. Round of applause, please.
Hello, everyone. I'm Tochi Chukwuemeka. I'm a second-year behavioral neuroscience and design major. So a little bit about the activities I do other than research is I volunteer at Brigham and Women's Hospital. I volunteer at a nursing home. It's called Cheryl House, and I'm an organic chemistry 1 TA. So I've been doing research ever since my first semester here, all thanks to the Dean's Undergraduate Research Scholars Fund. I was matched up with a lab that I really like. The PI is Dr. Praveen Kulkarni and the director is Craig Barrett. So over the course of the spring, I was involved in like 2-3 studies. It was all focused on the hallucinogen psilocybin. So we were feeding adolescent mice psilocybin and seeing their brain function. And then there's another study where we would bop the rats on the head to mimic a mild concussion, and then we would treat them with psilocybin. Oh, my future plans this spring, I plan on going on a co-op, and for the future, I want to go to medical school and become a neurologist. So using the Dean's Undergraduate Research Scholar Fund, I was able to do an independent study this summer. So we were extending our data and research from the previous spring study with the psilocybin in mice. So we were giving them fox scent, which mimicked punishment, and while they were awake in the scanner. And then we were giving them benzaldehyde to mimic pleasure and reward. Oh, I would like to thank Dean Siv, Assistant Dean Reinsberg, Fabiola, and Linda. They've really been a help, and I couldn't do this without them.
Thank you so much. Now we have our next student, Nicole.
All right, hello everyone. My name is Nicole Mongillo. I'm a 5th-year ecology and evolutionary biology major with a Spanish minor, and I'm on my way to graduating this December. Over my time at Northeastern University, I've been lucky enough to do 2 co-ops, one with Katie Lauderhouse at Northeastern, she's here tonight, studying oyster genomics, and one at Harvard University studying octopus chemotactile sensation, where I was also an animal care technician. Outside of my classes and co-ops, I am an active member of our campus's musical theater company. I'm also a teaching assistant for geographic information systems this semester and have been a TA multiple times in the past. And I recently started working at the new EXP Wet Lab Makerspace as a student worker. My research today will be focusing on exploring patterns of yield variability across spatial scales in Costa Rican coffee crops. And I conducted this research in Gabriela Garcia's lab. She's been a wonderful mentor. And my future plans are to complete my honors thesis as an undergraduate and graduate in December. Okay. And then to continue working with the Garcia lab on this research and to eventually pursue a PhD in marine genomics. So getting into coffee yield variability in Costa Rica, what we knew before going into this project were a few things. One is that coffee is a crop that exhibits alternate bearing. Alternate bearing is a phenomenon in some plants where they have years of very high fruit yield, followed by a year of low yield, followed by a high yield year, and so on and so forth. What's interesting about this pattern of alternate bearing is that the degree that yield can change from year to year is often in and of itself variable. So variability in variability, getting very inception. So that change can be either very great or very small, but it is hard to predict what that degree of change will be from year to year. And it is also very difficult to figure out what the drivers of such fluctuations are. We also know that these yield cycles, these years of high and low yield, can sync across different spatial scales. So all in all, we have a crop that can make it very difficult for farmers who are farming this crop and who depend on it for income. It's not very stable year to year. So what we wanted to know was, do alternate bearing, yield variation, and yield synchrony differ by geographic region in Costa Rica? And do we see these same differences at different spatial scales? In terms of Costa Rica is divided up into cantons, which you can think of sort of as counties, and then the coffee governing bodies of Costa Rica further group these cantons into coffee growing regions. So do we see the same patterns at the canton level as well as the regional level? And if so, why? What are the drivers of this? So to answer this question, we needed a lot of data. And I was lucky enough to visit Costa Rica with Dr. Garcia and some of our collaborators from Tufts in the summer. And we were able to speak to many different coffee farmers and coffee researchers who were extremely generous with their time and data, and they provided the data that informed this analysis. So what we found from these results, and this is an ongoing analysis, is first of all that we have yet to identify any major geographic or farming management drivers of alternate bearing. And what I mean by this is that there's no clear pattern between geographic features of a farm, such as its elevation, or management factors of a farm, such as whether it's organic or not, and its effect on whether or not we can detect alternate bearing. And this means that this cycle of ups and downs, this alternate bearing, isn't something that farmers can simply manage away. And when we spoke to a lot of farmers in Costa Rica, we heard many, many times that if you are a good farmer, you should be able to have a stable crop from year to year. And we're finding more and more that it's not that simple. Next, we also found that yield synchrony, so the synchrony of yield cycles, decreases with increasing distance between coffee growing cantons, which means that coffee growing regions that are closer together have more synchronous cycles of alternate bearing than those farther apart. And this tells us that farmers may be able to collaborate with their nearby neighbors to predict yield from year to year. Lastly, we also found evidence that a fungal outbreak in 2014 in Costa Rica led to increased yield synchrony at the canton and regional scale. And this was a really important thing to notice because these lasting synchronized fluctuations will mean that we now have a large population of farmers that will be experiencing economic instability all at the same time for some years to come. These are things to notice. Overall, this research is a really exciting opportunity to be able to study these patterns. We see these same patterns in managed populations that we do in wild plants and wild crops. And that's not necessarily what you would expect. You would expect that in a farming situation where variables are really kind of minimized, things like nutrients and water availability are really standardized, you would expect to see some of this variability in year to year go down and to change the patterns that we see between farms and natural plants. And that isn't the case. So we have this wonderful opportunity to look through all of the many years of farming data that we have to try to inform our understanding of the natural world, for which there is a lot less data than in our managed systems. So with that, I would like to give a huge thank you to the Halverstadt Family Summer Internship Program. Thank you for coming tonight, and thank you for this opportunity. It's been marvelous. I'd like to thank Dr. Garcia, who's here tonight, for being one of many wonderful mentors I've had at Northeastern. And lastly, I'd like to thank Ecafé and Gatier, which are two Costa Rican coffee research initiatives that provided the data for this project. Thank you so much.
Wonderful, wonderful. Oh my gosh. And next we have Charlotte.
Hi, my name is Charlotte Roth, and I'm a second-year biology and mathematics major. So I just wanted to mention some of the study abroad opportunities I've had through Northeastern. I was a global scholar last year, so I studied in Oakland and then London. So this is my first semester here in Boston. I also participated in a dialogue in Ghana where we worked in the health system for 30 days. For some activities that I'm involved in here in Boston so far are the Alliance of Civically Engaged Students, Northeastern's EMS program, the Women's Interdisciplinary Society for Entrepreneurship, which I'm continuing from London, as well as Red Cross Club and Irish Dance Club, and a few others. So my first research opportunity was actually working with the Jill Barrett Research Scholars Program this summer in Oakland in the Young Lab, and I'll be talking to you about that today, which is the influence of size in the cuticle on C. elegans running response to alternating current. And for some future plans, my first co-op will be next fall, and I'm also planning to attend medical school. So for the influence of size on C. elegans running response to alternating current, if you can see the pointer on this side. So the intention of this assay is to discover the molecular pathways and genes connected to emotions. In this case, a behavior akin to fear. So we're using a model organism known as C. elegans that are trackable. To perform this assay, we used 9-centimeter NGM agar plates, and we micropipetted 8 1-microliter spots onto the agar. And then we eyelash picked 6 young adult worms onto the plate, which can be seen up here. If we look to the left of the screen, you can see what we designed as the ZAP. So the ZAP is 30 volts of electricity with 4 hertz and 80 milliamperes. And to record this data, we record 30 seconds before the zap, 60 seconds of this zap, and then 30 seconds after the zap. What we decide as a leaving response is when the worm is at least 1 worm length out of the lawn and moving in the direction away from the lawn. As you can see in this photo on the bottom left, this is how we measure the worms. So we take photos of the worms. In this case, this is our wild type, and we use a program known as Fiji. So in this video on the top, you can see a response of our wild type worms, or our N2. When the zap is administered, they begin to increase speed and leave the lawn, as seen. And for the worm video on the bottom, this is of small 1 RU18, which is a gene that was identified through a forward genetic screen to have a defective zap response, meaning they leave the lawns at lesser rates. And they have many phenotypes that are different from wild type, but we decided to focus on 3, those being the cuticle, excretory canal, and size. And for size, which I'm talking about today, they're smaller than N2 typically and dumpy. So for our results, you can see that we're comparing length with the percentage of worms leaving the lawn, with our error bars being standard deviation. For N2 at the top, you can see they're one of our longer worms, and they also have the highest percentage of worms leaving the lawn. And for small 1 RU18, you can see they have a lower percentage leaving the lawn, as well as them being shorter in length. And for some final acknowledgements, I would like to thank the Barrett Foundation for running this program, especially this summer, as well as the Mills College at Northeastern STEM Unit, the CGC, the Kimura Lab in Japan, as well as Dr. Jared Young, my PI, and all of the Young Lab, both past and present.
Thank you. Right, and we have our next student, Zach.
Hello, everyone. I'm Zach. I just graduated in May of 2024, and I'm currently in 3Cs, which is the Marine Biology Master's Program. My co-op, I worked down at the Marine Science Center at OGL, which is a biorepository that stores marine tissues. But thanks to the Jill Barrett Research Scholars, and the Vollmer Lab, I was able to work on coral genomics. What I enjoyed doing is I did rock climbing, husky hunt, a whole bunch of different activities that we could get into later. My research that I'm focused on today in the Vollmer Lab was from video to data, where we used random frame sampling from underwater video to accurately determine reef health. In my future plans, I'm gonna get my master's and hopefully further the research that I'm about to tell you about. what is the Vollmer Lab? The Vollmer Lab are very specifically focused on a reef building coral. The reef building coral that they're focused on is Acropora cervicornis, also known as staghorn coral. There was a disease called white band disease. There is a disease called white band disease. It was discovered in 1970, and since then it's killed over 95% of staghorn coral. So in the Vollmer Lab, we're trying to figure out what causes it and why it seems that some coral reefs have a natural genetic benefit and are able to survive the disease better than other ones. So that's kind of where I came in. In order to figure out which reefs are healthier than others, usually you send a scuba diver down and they lay out a transect of usually 50 meters or so. And every meter they'll count, oh, here's some dead coral, here's some alive coral, here's some that's been affected. It's kind of time intensive and you have only a certain amount of time down there. So what the Vollmer Lab usually does is they'll take a video, as we can see right here, hopefully it plays, where you're going along and recording the whole reef. You'll go 50 meters down and back. The thing is while this gives you somewhat accurate way of telling how healthy your reef is, it's pretty time intensive. And my job and my project was to make this a bit more efficient. So I developed an application that instead of using transects, you can just record videos of the entire reef, and it will, you plug the video into the app, and it will give you random frames from the whole video, and then you go in and manually select alive or dead, and it allowed for higher quality data, and it significantly improved it. And so this way we can now determine which reefs are healthier and which reefs we can now look further into the DNA and the genetics of to figure out what we should study. And so a huge thanks again to the Jill Barrett Research Program. Without them, this was my first real research opportunity, and it just meant so much to me. And I also want to thank Dr. Stephen Vollmer and everyone in the Vollmer Lab for mentoring me and helping me out. So thank you so much.
Thank you, thank you. And we have our next student coming up, Bita.
Hi, I'm Bita Adel-Zadeh. I'm a fifth year here at Northeastern, and I've been studying biochemistry. My minors are data science and behavioral neuroscience. I've had the very fun privilege of doing 3 different co-ops, all in very different areas. My first was at a startup called Full Circle Therapeutics, where I was part of their discovery biology team for genome editing. My second co-op was with Cerevel Therapeutics, and as part of their in vivo pharmacology team, I worked with central nervous system relevant preclinical models for Parkinson's disease. And currently, for my third and final co-op, I'm in Dr. Lei Liu's Laboratory of Neurology at Harvard Medical School and Brigham and Women's Hospital. He's here today also, which is very fun. And I'm very thankful to Carol Shapazian for my sponsorship for this work this semester. Apart from the research that I've done on co-ops, I've been a very active member in Dr. Rebecca Shinsky's Laboratory of Neuroanatomy and Behavior. A highlight of this experience was receiving the Summit Peak Award, where I got to study the effects of pregnancy on associative learning in rats. And when I'm not doing research, I love to be outside and bake. I'm from California, so this is a photo of me hugging a tree in Yosemite this past summer, and a couple of very fun things that I've baked over my time here in college. For future plans, I'm very happy to share that I will be continuing in Dr. Liu's lab for my biochemistry capstone next semester for my final semester at Northeastern. I'm also currently applying to neuroscience PhD programs for the fall of 2025. So in Dr. Liu's lab, we primarily focus on clinical biomarker research for Alzheimer's disease. And so specifically, the work that I'm doing is characterizing tau species in plasma. So for background, Tau is a microtubule binding protein whose aggregation has been widely associated with the progression of Alzheimer's disease. And most established biomarkers for Alzheimer's disease are found in the CSF. However, collection of the CSF is a very invasive and painful procedure for patients. And so there's a clinical need to develop a blood-based biomarker detection method to allow for a less invasive way to monitor the progression of Alzheimer's disease. However, this is presented with a challenge, which is that plasma is a highly enriched and complex solution, which makes the detection and identification of tau species very difficult. And so the work that I've been doing for the past few months has been developing an innovative chromatography technique to separate tau species and then using an ELISA method to quantify specific tau epitopes that are present in our fractioned plasma samples. So you can see this is full-length tau, human tau, and tau will cleave, it'll aggregate. And what we're interested in is knowing what portions or what epitopes of tau are present in our peripheral system. So leveraging the biochemical properties of tau, we're able to isolate various tau epitopes that are present in plasma. And then we're asking the question of what parts of tau are really, what are we seeing? And so we are using various different antibodies that will bind to different regions of tau to detect how much tau is present in our fraction samples. And so far, we've seen a greater detection of tau epitopes of our tau epitopes of interest in plasma than in the CSF after our chromatography technique. And this suggests that the amount of tau in our system is heavily underestimated. So I'm very excited to continue. There's a lot of work left to be done, and I'll be continuing on next semester. And I want to say thank you again to the Shapazian Research Co-op. And also, I don't think Katie Dioli is here, but she has been a fabulous co-op advisor she connected me to Lei. And just over the past several years, I'm very thankful for all the opportunities I've been able to have. So thank you.
Thank you. And now we're introducing our next student, Jasper.
Right, hello, my name is Jasper. I'm a recent graduate from Northeastern just 3 or 4 weeks ago in August. I graduated with a major in psychology, a minor in ethics. I did have the privilege of having 2 co-ops at Northeastern. My first was at the Attorney General's office as a community engagement program assistant, and my second was as a substance abuse case manager at Pine Street Inn, both of which I loved. From my activities, I was a very active member of the Buddhist group on campus, the Interfaith Social Action, which was essentially a volunteering organization, and the Choral Society. The research that I'm going to be talking to you guys about today, we studied moral emotions and activism about climate change, and I did that with the Sustainability and Social Change Lab with the PI Sarah Constantino, and that was supported by the Paul and Grace Ward Martinez Research Fellowship. In my future plans, I'm hoping to get into some clinical work. I got a job offer today from McLean, so we'll see how that goes. Thank you. So for this project, our main question was figuring out what causes climate change related burnout, because we see that there are quite a number of people who care about climate change, a lot of people who have hard feelings around climate change, and not as many people taking action around climate change as there are people who care about it. So we want to figure out where's this burnout coming from. To do so, we use this concept called moral distress, which comes from the nursing literature. Since the 70s, there have been studies that have documented nurses who have over time built up this sort of emotional tension from seeing some sort of moral wrongdoing or not being able to move towards treating their patients in the way that they would deem morally appropriate, because what they would like to do was overridden either by directives from doctors, which took precedence, or by some sort of institutional constraint which prevented them from doing what they would consider morally right. Exposure to those sorts of stressful moral situations over the long term led to things like burnout and disengagement. And we wondered whether a similar phenomenon was happening in environmental spheres as well. So we broke down moral distress into a few theoretical components, including a moral foundation, which is like seeing the sorts of moral rights and wrongs relating to the environment, a lack of agency, and negative emotions around climate change. We found, well, we looked in both activists and in a general population, which we broke down into Democrats, independents, and Republicans. And we found that activists had the highest level of moral distress, followed by Democrats, independents, and Republicans, which was interesting. It actually went against our initial hypothesis. We had hypothesized that activists would have a lower level of moral distress because they are the ones taking action, and that might reduce some of the tension that they feel. But it seems that the sorts of activism that they were participating in alone were not enough to reduce their sorts of the morally hard feelings that they were experiencing. When we broke it down to its theoretical components, we found a very similar pattern among most of the dimensions except for agency, where everybody who we surveyed had a similarly low level of individual agency when it came to addressing problems with the climate. For future directions, I would love to look into seeing whether we can adapt some of the solutions, some of the interventions that the nurses have developed to address moral distress and bring those over to the environmental world as well. Thank you guys again. This was supported by the Paul and Grace Ward Martinez Research Fellowship, for which I'm very grateful. It allowed me to focus and spend so much more of my time on this research project. Thank you guys.
Thank you. And for our next student, we have Saad.
Hello. Sorry, I have a very quiet voice. I'm Saad. I'm currently a third year. My majors are bioengineering and biochemistry. Basically never been relaxed. I'm on my first co-op at the Monaghan lab. I've kind of been annoying James, who's our current chair. Some of my hobbies, I do botanical studies, drawing, like to read. My research is nerve dependency of limb regeneration in axolotls. I can't pronounce the scientific name, but it sounds sciencey. Keep it there. My future plans is to continue the research of regenerative medicine. It's a really new field. So axolotls, they kind of look weird, but they're a species of salamanders. One thing that's unique about them is that they're neotonic, which means they stick in a juvenile state. In us, we tend to grow old, we age, but axolotls tend to keep the same regenerative aspects of development throughout their entire lives. They're able to regenerate nearly every major organ, their spinal cords, their limbs, even parts of their brain. Right now, we're trying to investigate nerve dependency of regeneration. We found that when you remove the limb or when you remove the nerve, limbs don't regenerate, the parts don't grow back. In us, when we get an amputation or when we get wounded, we develop scars, but with axolotls, they're able to regrow it back, and that is due to the nerves. James's lab has worked to identify neuroglin-1 as a key molecule. They found that in limbs that have lost nerve supply, they're able to rescue regeneration. Research points towards the dorsal root ganglia, which is located in the spine, as a source of these neurotrophic factors. My current research it's currently developing, is to implant these nerves, see which factors are released, how they influence different parts of mechanisms. Right now, I'm trying to look at the immune system, because again, it's shown to be a very important part of wound repair and regrowth. The impacts of this is that if we're able to understand how axolotls are able to do this, we can build new therapies for regenerative medicine, things from growing limbs, growing damaged organs, and there's a lot we can do with it. I'd like to thank Robert E. Matz. Not sure what he looks like, but thanks to him, and also James Monaghan for taking the time to mentor me and take me on. So yeah.
And next, we have someone I know, Migue, of course. Welcome up.
Hello, everyone. My name is Migue. I am a fifth-year cell and molecular biology major, and I'm in the honors program. I'm set to graduate in May of next year, which is crazy. I'm also looking for a job, so, connections. So I have done 3 co-ops. I did research at the Greenberg lab studying immediate early transcription in neurons. And then I worked as a Schafer research scholar at Dr. Winchai's lab, where I looked into cell communication within bacterial biofilms, which are these communities of bacteria. Right now, I work at Moderna, and interestingly enough, I'm also looking into biofilms. So after this, I am actually presenting some of my research from the Chai Lab in 3 national conferences. I'm thinking of, I just submitted like 2 abstracts to like 2 conferences. And after that, I'll be applying to fellowships, jobs, and MD PhD programs. So this is a schematic that I made of all of the research that I did in the Chai Lab. Biofilms are ubiquitous in nature. They are found in soil, they are found in hospital catheters, and they're also found in your teeth in the form of plaques if you don't floss hard enough. And basically what I did research on was this molecule known as bicarbonic acid and how it is a vital cell signal between bacterial cells within biofilms. And so biofilms, why study biofilms in the first place? Well, first of all, they are protective. They are notoriously resistant to antibiotics. And so if you keep adding antibiotics to them, they just don't go away. And so finding ways in which they form will prevent their formation, thus preventing them from ever being targeted by antibiotics in the first place. And so this research wouldn't have been done without the support of my mentors, especially Dr. Win Chai, who I think is the best PI in Northeastern. He has supported so many undergrads at Northeastern, and they've just gone on to accomplish so many things. And I've been so lucky to work alongside Ying Hao, Nicole, Dr. Leticia, and especially Jillian. And they've been the greatest people that I've ever known. And I'm so happy to be part of this lab. And of course, I'm so thankful for Veronica Porter and Dr. Andrew Schafer. He visited me this past April, and we were able to talk about medicine and basically his book, Unclean Hands. And he gave me a signed book copy. So yeah, nice meeting you all.
Wonderful. And for our next student, we have Zach.
Hello, great to be here with you all. I'm Zach. I am also a recent graduate from our undergraduate program. I got my Bachelor's of Science in the honors program through the mathematics department with a minor in physics. Some of the research that I started while I was an undergrad here was 4 semesters leading up to my honors thesis, and that was on cluster algebras under Professor Milan Yakimov. And the program which I will be talking about today is the Northeastern Summer Math Research Program, which primarily is a summer research experience for undergraduates that runs every year in the department, of which I have participated 3 times. And that is partially funded by the Dean's Fund of the College of Science, of which I am extremely grateful, as that allows us to broaden the opportunities to more students in that program. Some other things that I got up to in undergrad. In my department, I was pretty active. I served on the e-board for our math club for 2.5 years, with the final year being as the club's president. And I was also active in mentorship through our Math Engagement Mentorship Association, an undergraduate teaching fellow, and outside of my department, I was, and hopefully still will be, active in the New Stage Musical Theater Company on campus. My future plans, I'm starting my PhD in math here at Northeastern this semester. So I guess, hopefully, that will go nice and smoothly toward a career in math research. So what I wanted to talk about today is the Northeastern Summer Math Research Program. And like I said, this is a summer program. It runs for 2 months during our summer 1 term every year. And I did 3 projects, of which I'm going to be talking about the most recent one. So this was a group project with 2 graduate mentors, Sean Carroll and Vitor Gulis, and some other undergraduate collaborators, Jonah Marcus and Jordan Martino, with funding through the NSF, the department itself, as well as the College of Science Dean's Fund. So to give you a little context for what this work was on, like I say here, it's an exploration of categorical structures. And what does that mean? As Dean Siv told you all, math can be described as the language of the universe. And one language that we use to describe math is something called category theory. And what I have here as figure 1 is a minimal example of a category here. As you see, we have some objects, A, B, and C. We have maps or between them called F and G, and we guarantee that we can always compose those maps, as you see with the little circle there, G composed with F, as a map that always exists between them. And this technique of describing objects and maps this way was first developed by Eilenberg and MacLean in the early 1940s. So this is a pretty long-standing way of describing mathematical objects, and it's pretty fundamental to areas in math like topology. So for those of you who don't know, the study of invariant properties of shapes when you're allowed to deform them. And that has also found a lot of use in computer science and physics. But what I'm talking about today is kind of a variation on category theory in something called higher category theory or higher homological algebra. And this is a very new concept. The concept of this n-abelian category is what I'll be talking about. Was only developed within the last 10 years. But some of the research that's been involved on it has been going on in our own department under people like Professor Gordana Todorov, as well as Vitor Gulius, my primary graduate mentor for this project. So this project was actually a continuation of work he's been doing toward his dissertation. So given a category with the properties we want, we call something with kernels, and I give you a picture of a kernel there. So imagine that a kernel is that set of maps with only one map, so n equals 1. What we are generalizing to is a category where instead of a abelian category that has kernels, we want an n-abelian category with these n kernels of arbitrary length. And so what's interesting is that despite this idea having started to be explored over the course of the last 10 years, the first example of these categories actually existing is in the form of something called a higher Auslander algebra. Now, Auslander is a local mathematician. He taught at Brandeis for most of his career, and he first constructed this Auslander algebra back in 1974. And this higher homological analog is only discovered back in 2017. So very, very new. And we were hoping to look for another example. Because so this first example was in 2017, we were hoping to find any other way to produce these types of categories. And so what Vitor had started was he found a correspondence between properties of an object called a ring and and you could take a ring with certain properties and map it into a category that we'd like. So this project said, how can we make a bunch of these objects that Vitor found a correspondence with? And what we did was take this process called generating group rings to create a bunch of possible examples that we could then test. And what we found actually was that the vast majority of these objects could never ever possibly give us what we wanted. So this project, sure, we set out with the goal of finding some examples, but we actually ended up showing definitively, we proved that in any case of a finite group or finitely generated group, that no such example was possible and essentially ruled out the entire possible class of categories, which was very good and significantly narrows the search of possible techniques to find if there are any other ways to construct these objects. But yes, I'd like again to thank Vitor for the work that led to this project being possible, as well as the wonderful Northeastern Math Summer Research Project, because this program allows research and targets research toward those students, early undergrads who have not had a lot of access to those opportunities, and is a great way for students to first get engaged in research, especially in a field like math where there aren't as easily available things like labs. And I'd like again to thank the Northeastern College of Science, specifically the Dean's Fund, for the contributions to that program. Thank you.
Thank you. And before we end off, I just want to thank everybody for being such an attentive audience and all of the students and the staff that put this together. Thank you all. You guys are working really hard, really smart people. And as we see, research is interdisciplinary and with the goal here at Northeastern to solve the unknowns of the world. So thank you all for being here.